Well, that's a different approach.
Come back. So it is my pleasure to be able to introduce John George to our next talk, who I actually worked for at Los Alamos after I did my undergraduate degree. So it's very exciting for me to keep running into John, who's now deputy director, right? Deputy?
Right, group leader.
Group leader at Los Alamos. So,
All right. Well, first let me say that I have to congratulate the organizers of this meeting because seeing from the inside the kinds of problems that DOE has posed for people organizing meetings recently, a couple of months ago I thought there was virtually no chance that this would happen. So my congratulations to the organizers for what I believe has been a very interesting and engaging meeting. So I work at Los Alamos National Laboratory. I came here after working at the National Institutes of Health for several years, where I was working on problems of vision at the very front end of vision. How do photoreceptor cells work? I came to the study of biophysics and physiology, though, from computer science. Back in really the dark ages of computer science. But the thing that motivated me all along was the possibility of trying to understand how the brain processes visual information. It seemed to me that that was a paradigm for really understanding how the brain works. And that's what we'll talk about today. A lot of my career has been focused on studying how vision works and how animals and humans do vision in a wide variety of systems, and developing technologies that allow us to study that, especially with a focus on studying large collectives of neurons. But in the last few years, our focus has shifted, really driven by the idea that computational methods may be the tool that ultimately allows us to understand how the brain works. There's only so far, I believe, that we can go with experimental methods, and ultimately to crystallize our understanding of how the systems work, and to show that we in fact do understand how they work. It's necessary to go to computational systems where we can reproduce the function of the systems that we're interested in. This leads us to the idea that not only do we treat this as an enterprise to try to understand how the brain works, but that ultimately we treat it as an enterprise to build systems that emulate brain function. And we believe that, that neuromimetic systems not only have big advantages from a cognitive point of view, that we can do things and understand things about images that we can't do with the present systems that we have, but that ultimately it allows us to have systems that can work in real time, that they can work on real world data, that we can achieve systems that are portable, low power, massively parallel, fault tolerant, adaptive, all of the things that are important if you want to put a visual brain out on a piece of hardware that's deployed, or alternatively if you want to build a supercomputer that doesn't bring down the whole power grid, or that is not completely unworkable because of the mean time between failure. So what we really focused on, or are focused on, is trying to understand how we can take these devices, which are not deterministic, they're probabilistic, and how we can use redundant, specialized computation with pieces that really aren't that great to achieve computational and cognitive performance that we can't touch with even our very impressive current day supercomputers. So the elements of neural computation that we think are important are summarized here. I would argue that that Hopfield and a lot of others really did get it right, that connectivity of neural systems is probably one of the most important keys to their functions. And this is not universal all-to-all connectivity, even though that is something that neural systems can achieve by virtue of specialized wiring schemes. There is dense local connectivity. There is dense feedback, and this is both lateral and between functional areas in the brain. It's clear and increasingly clear that this is an important aspect of how the systems work. An important aspect of how cortex organizes is that there are specialized areas that have particular functions. And realistically, these probably evolved from other areas. That's the way biology works. It takes a function that sort of works, it replicates it, and tries to use it for something else. It has to do that because it can't afford to design from fresh. It has to keep whatever is working because otherwise it's not going to survive. But it can take those things, reuse them, recycle them, and do new things with them. And I believe that's why we end up having 30-some or 3 dozen visual areas that are specialized for particular kinds of computation. I think another key idea that is important in biology and in understanding brains is the idea of feature detectors, that cells in some sense do represent some pattern in their input space, some spatial and temporal pattern in their input space. An extension of this idea that's been talked about some at this meeting is the idea of sparse over complete representations, that we have that a set of neurons, say in a cortical column, represents a set of basis functions that can represent a little bit of the outside world. But it's not a set of basis functions like you might get from principal components analysis or SVD or something like that, because there's no requirement that these be orthogonal bases. We can have multiple ways of representing the same bit of the outside world, and there's no real penalty for it in biology. Another thing that I think is important is that neural systems tend to encode change, and they tend to encode anomalies. So as long as everything is as expected, the system doesn't really have to deal with it very much. But when there are things, anomalies and changes, that's when the system has to go to work. There clearly are learning and STD rules that allow the systems to learn to detect patterns in temporal relationships and causality. But all of that aside, and that really has been the focus of computational neuroscience probably for the, well, I guess close to 30 years now. The thing that I think has been missing, and the reason that the systems that we build, like HMAX, are still orders of magnitude less good at cognitive tasks, is because most of these systems neglect the fact that neurons spike. And part of my point today is to try to make the case that this is something that we have to consider, that it gives us power well beyond a simple way of representing data. It also makes things much more efficient. It makes communication in biology and in systems a lot easier, but I would argue that, that this is the thing that's primarily missing. And it's a little surprising because this is where the modern era of neuroscience starts. Hodgkin and Huxley, their claim to fame was that they were able to describe in a set of differential equations why it is that a neuron spike, how it is that that a set of a few ion channels can cause this system to produce this this delta function-like transient that that encodes information in a useful way. So just a little bit of neuroscience, since not everybody in this audience is a neuroscientist, and this is very basic. I would say that biologists tend to revel in the detail, And there is a tremendous amount of detail that goes into a neuron. Some of it's clearly more involved with how neurons develop and how they survive and how they metabolize and all those things. But the part that I think is most essential for information processing can be relatively discreetly summarized in the form of kind of a leaky integrate and fire neuron. What this means is that as inputs are received, that charge on the membrane builds up, that if you don't do anything, that charge gradually bleeds away. That's the leaky part. But that if a threshold is reached, then the neuron fires, and then the charge dissipates. I would argue that that really is the essence of neural computation, and it's how you use that that accounts for what neural systems can do. So a little bit more about this in cartoon form. One of the ideas that is kind of key to neurophysiology is that if there is some driving input, if there is some stimulus from the outside world, or if there's some drive from other neurons, what that causes is an upswing in the membrane potential, or a downswing depending on how you look at it, but it is a tendency for the membrane to depolarize, and if it reaches some threshold, it fires, produces a transient, and so forth. Now, in the lower frame is the extension of that idea. If you have continuing input, or if you have stronger input, first the cell is likely to fire quicker. It's likely to shift its phase relative to the onset of the stimulus. If you have continuing input, what's going to happen is that this cell is going to fire again. And if you have stronger continuing input, it's going to fire at higher frequency. And I think a lot of what we think about neurophysiology comes from measurements of this kind of phenomena, and led us to the dogma that I was told when I was in graduate school, that information is encoded in the rate of firing. And so this is one example of that. You've got an oscillatory stimulus, you have a little burst of firing. After the system reaches some threshold, the strength of the stimulus is encoded in the rate of firing, as represented in these peristimulus histograms, and everything works nicely and can be thought of as a sort of linear system. This gives you a measure of neural activation that is a scalar value. And if this is all that the system does, I would argue that you don't need spikes. You can represent the same kind of information with some sort of scalar activation parameter, and things work just fine. And it turns out that you can do a lot of what neural systems do with exactly this kind of computation, and you really don't need spikes. But spiking takes you a step further. So one thing that spiking does is it gives us a way of integrating the fact that the inputs are also coming in as spikes. So what happens is that you get an input from a cell that is talking to you. You produce a little blip in your membrane potential. Nothing else happens. It goes away, and you're back to baseline. Another spike comes in. The same thing happens, and nothing happens. You don't produce an output. On the other hand, if I have two input spikes that occur within a short amount of time, then it causes this thing to go super threshold, and the system fires. This means that one of the fundamental things that neural systems can do, or that neurons can do, is act as temporal integration devices, so that they can, if spikes come in within a certain narrow window of time, they're much more likely to produce a spike. And one consequence of this is that neural systems are exquisitely sensitive to synchrony and coincidence. When spikes arrive at about the same time, they're much more effective than if spikes are spread out randomly. Now, you can imagine that if you have random spikes, but you change the frequency, you're going to increase the probability that they're going to arrive within a narrow window. And so, even rate codes do do mean, if you have high frequency firing, that you're more likely to produce an output spike. But it also means that if you have temporal structure beyond that imposed by frequency, that you get a computational benefit. Another thing that I'll mention is that there is a possibility, and there is observed demonstration, that there are subthreshold processes. Neurons are connected together as a big resistive network. Now these little responses from cells that are connected resistively tend not to be enough to cause the cell to fire. That would be a bad thing for a computational engine. But what it does tend to do, what it tends to mean, is that the activity of all your neighbors influences the probability of your firing. And one of the things that can do is to produce a local clock signal that tends to synchronize firing within neural populations. This has led over, I would say, the last 20 years to the idea that the temporal code in neural firing is what is important. And I think this is the poster child example of that kind of process. This is from some work by Neuenswanger and Singer, who I would argue is probably the person who's most effectively pushed this idea. So what they did was to measure from a couple cells in retina. It's a very simple case. And what they find is that they put a spot of light or a bar of light on one cell. It produces something that tends to oscillate. This is an Autochlorellogram, and it has the characteristic structure that's suggesting, that's suggesting that this is an oscillatory response. And if you look at the other cell nearby that's responding to a different spot of light, it also has an oscillatory response. If you look at the cross-correlation between those two cells, there is no significant cross-correlation. On the other hand, if he stimulates these two cells with a bar of light so that they're both stimulated by the same structure, what you find is that both of the cells are oscillating, but you also find that there is a strong cross-correlation. So these cells tend to synchronize. They tend to synchronize their oscillations when they're co-stimulated by a structure. Now this kind of observation has been made not only in retina, which is where this comes from, but certainly in the lateral geniculate nucleus, in cortex, and more recently even between visual areas. So it really seems to be a key idea that neural systems use to encode information. That you have a rate code that encodes activity of my own little patch of the world that I'm interested in looking at, but I have this temporal synchrony code that encodes my relationships to other neurons that may be looking at a different part of the world, but if we're looking at two parts of the world, or if we, for instance, are both looking at an edge with the same orientation, and those edges happen to be collinear, chances are we're going to be oscillating in synchrony. And the fact that we're oscillating in synchrony conveys the additional information, the meta information, that we're both part of the same edge. And I argue that that's where you get the additional dimension of representation in this system. You get it from the fact that these patterns across ensembles of neurons really can encode meta information. It really is encoding complex information rather than scalar information. So, so why do we use temporal codes? I would argue that probably the reason that they started is because there is a lot of structure, temporal structure, in the environment. So if you're an animal and you're sniffing, the little burst of odorants come in little bursts, and that tends to synchronize the firing of those things. But alternatively, if you have an edge that's moving across your visual field, that's going to tend to produce synchronized activations of series of edge detectors in your visual field, and that physical or that aspect of the physical environment gets very naturally encoded by the temporal code. I think the fact that there is this evidence that that metadata is also encoded by temporal codes is the fact that the system said, well, here's a feature, what can I do with it? And I would argue that that ultimately temporal synchrony and synchronized oscillation codes become a way of encoding information, encoding relationships between disparate information that have no physical temporal connection. It becomes a mechanism of representing information. Now, a lot of people say, well, it's a epiphenomena. It's, yeah, we see it, but what does it mean? And this has been a difficult problem because it's very difficult to show that the brain actually uses the information that's clearly present and encoded by these kinds of temporal structure, these kinds of temporal dynamics in the response. But there are some examples. This is some work by Ishikani and collaborators, and what they showed is that there's a particular kind of cell in the frog that responds to a looming stimulus. This is a big dark shadow that's getting bigger, right? And if you're a frog and there's a big dark shadow that's getting bigger, chances are you should do something because otherwise you're going to become food. On the other hand, if there's a little dark shadow that's buzzing around, chances are that is food and your response needs to be different. So what they showed is that these dimming detectors do have this characteristic of synchronized oscillation, but that if you block the synchronized oscillations by pharmacological manipulation, that you can block that behavioral response of a frog, which is to normally jump away if it sees a big looming shadow. There are also examples from human neuroscience that suggest that gamma band power, this is 40 hertz to 80 hertz kind of power in humans, is associated with accurate cognitive responses in psychophysical or cognitive tasks. So there is at least the beginnings of evidence that this gamma band power is used for cognitive processing, is used to encode and extract information about the things that we're looking at. So, so moving on, that really is, I would argue, where the advantages of spike-based processing come. It allows us to encode a richer view of the external world, hopefully a lot richer than things like than abacus or fingers or ultimately even than things like digital electronics, where we still tend to represent the external world in terms of arrays of scalars. But I would also argue that because of this characteristic of being able to do temporal integration, that neural systems are exquisitely tuned to detecting synchrony in their input field. And in fact, that is the thing that makes them useful. So I wanted to talk about some examples that kind of apply these ideas. I wanted to go back to a concept that I mentioned earlier, that the world tends to be represented in terms of basis functions. And in the visual world, what this means is that we take a small patch of a visual scene and we try to abstract it into patterns of light and dark. And if it's time-evolving visual information, such as in video, we also have an element of temporal, of time-dependent receptive field behavior. So what we do is expose systems to a large number of exemplars of the kinds of things that we see in the visual world, and what we wind up with is a set of basis functions, a set of a sparse over-complete representation or a dictionary that allows us to represent that. Now, if we then give it a new patch of arbitrary visual pattern, what tends to happen is that this gets represented in terms of one or a few of these feature detectors or basis functions. And this allows us to represent the visual world. Now, I'll come back to this idea in a minute, but moving on to application, we were involved in the last several years in a project that was sponsored by DARPA called Neovision 2, and we sort of got in the back door, but and largely because we proposed that spikes were important and that we should explicitly deal with them in computation. What we actually started with, though, was something that is agnostic with respect to spikes, which is the HMAX model that Peugeot and Sarah and colleagues have developed. What it really captures is the idea of basis functions, the idea that there are hierarchical systems that process visual information and extract different kinds of representations from it. And in that sense, it's a neural-inspired hierarchical system. We certainly incorporate the idea that there are specialized inputs channels for color and for motion and even things ultimately like covariance. So we worked with these kinds of systems. Now, we started out working with the vanilla version of this system, but pretty early on we decided that the dictionary structure that the original HMAX model was not sufficient. So we very quickly moved to learned dictionaries. That bought us a lot, but ultimately we moved into what we call, or what people call, deep generative systems, and I'll describe a little bit about what that means. But on to what we actually did. So we were looking at overhead video. This is really what they're interested in, is to deal with the kinds of video feeds that you get from UAVs or helicopters or even satellites ultimately. So we took an original input file, and these are typically in the form of video clips, and we ran it through 3 or more modules. We ended up using these 3. One of these we applied to what was essentially our version of the HMAX algorithm, was with a bit of hot rodding that we did to it to deal with more appropriately structured dictionaries and to deal with generative models. We also use something that we call Petavision. This is actually a spike-based system that is intended to capture some of the other things that neural systems do. And in particular, in this application, we were using it to say something about shape, to talk about relationships between detectors, which is to a large extent neglected by the HMAX model. And then we also incorporated something that we called Petamotion, which is a motion processing system, actually not nearly as sophisticated as the one that we heard in the last talk, but that nevertheless worked with the ideas of motion energy and optical flow. We used these, we combined them. The back end, I have to say, the combination was pretty hacked. We tried different ways, but basically we were using conventional computing modalities and algorithms to try to combine these in various ways. With PAN, we were basically looking at color and texture. So we trained it on natural image patches. We learned a basis function. Ultimately, we moved beyond just the idea of these libraries that were learned from input and went to the idea of generative systems in which we have dictionaries. We can use this dictionary to describe an original image patch, but we can also reconstruct what it is that the system sees. So taking these basis functions and kind of inverting the process in a generative model, we can reconstruct the image. Another thing that we can do with this is to try to say something about the background, because I think most of us realize that not all pixels are created equal in our visual field. There are some things that are background, there are some things that are object, and we don't have any real difficulty discriminating between the two, but we found that most computer vision algorithms actually do have a problem. And using this with PAN, which actually stands for Petascale Artificial Neural Net, basically just a feed-forward network, we're able to get a lot of detections, but we also get a lot of false positives. With Petavision, on the other hand, what we're really trying to do is to deal with this problem of how do we detect things that are objects? How do we define objects? This is an example of a toy problem that we use to explore that. We call these amoebas, and the idea is that most humans, even presented with this kind of display for for 30 milliseconds, have no problem telling you whether there's a closed object that's being shown. You can make it more difficult by masking, and it turns out that there are characteristics that make mask more effective. But nevertheless, we have an ability to have this kind of object pop out. We tend to ignore all the distracting clutter, and we pull out the thing that is important. The formalism that we arrived at to describe how that system works is what we call, what we ended up calling ODD kernels. This is object distractor difference kernels. And what this really describes ultimately is the pattern of lateral connections that describe the connections from the side, the differencing types of connections that allow us to say whether this is something that represents a continuous curve or this is clutter that we should ignore. And I won't go into how we actually develop these kernels, but the net effect of using this kind of analysis on the imagery that we were working with is that it actually tends to eliminate a lot of the false positives. And so if we combine that and, for instance, the information that we get from motion, by combining them, we found that we could significantly increase the overall performance. Now, I have to say that, even in this kind of system, it's still human perceptual performance that is the gold standard, right? These systems don't do anywhere near as well, but they actually did as well or better than any of the other people out there, and I would argue that at least some of what allows them to be effective are the elements that we get out of biology. So I wanted to talk a little bit more about this idea of pop-out because it comes up in a lot of context. One of the most important contexts, in my opinion, is for segmentation. So how do we divide up the visual world into objects? Because we don't see pixels. We really do see objects, and we know that there's a background there, but we tend not to focus on it. We developed a number of kinds of models that accounted for this. This was actually from some work that is very theoretically described in terms of differential equations, only loosely connected to biology. But what this system does is to exhibit an ability through a series of relaxation steps to get rid of, to suppress the background clutter information and to leave the things that represent the continuous connected objects that we argue that we're interested in trying to perceive. Now, I don't really think that neural systems work in this relaxation mode, but the way that they do work may not, may be, well, it's clear that the way that they work can be mediated by mechanisms that could also mediate relaxation. So in order to try to be a little more quantitative with this work, we actually did a series of cognitive and behavioral studies. We presented human subjects with things like this. So we have in some instances we have a closed object with clutter. In other instances we have just clutter. And the task for you, if you're in this kind of experiment, is to report whether there isn't a closed object present, if an amoeba is present, but also to report your confidence. And it turns out that this is a very important aspect of this kind of experiment because it turns the response from a binary variable into a continuous variable. So what we found is that if you do this experiment with enough humans, you can characterize the response functions, you can characterize the receiver-operator curves and all that sort of thing. If we built models based on this so-called ODD kernel that were specialized to try to pull out closed figures, amoebas, first we get a description that is actually very similar to something described by Zucker and colleagues at Yale. which is what they describe as co-circularity. The idea that if neurons are representing edges that could lie on a continuous circular surface, that they tend to be connected. They tend to support each other. Whereas if they're on trajectories, if they are at angles that would not generally be part of a continuous closed object, they tend not to make connections. And so this gets represented in these kinds of connectivity patterns. I'll also say, obviously, though, that humans have evolved to represent corners, and so there are probably also, in addition to co-circularity connectors, there are probably corner connectors that are specialized to see the conjunction of a couple of different edges. But in any case, if we build models and we compare it to human psychophysical performance, what we see is that we can characterize human performance in terms of, for instance, the parameters of the clutter field, and we can apply these same kinds of perturbations and clutter to the model, and what we found is that the human and the model actually had a very similar dependence on, say, the radial frequencies of the clutter that suggested that, at least at a system level, it was performing in a similar way and giving us some confidence that maybe the underlying mechanisms of that performance were similar. So this is pretty gratifying, and this led to a paper in PLOS, and it motivated actually one of the responses that I made earlier in the meeting, that if we have systems and we're trying to understand and we're trying to debug things about the low-level details of the system, sometimes all we really have is evidence of how that affects the higher-level behavior and higher-level function of the system. So one of the things, though, that was very appealing, because, as I pointed out, we could do all of this stuff with relaxation-type models where, there's an initial burst of activity in the system, but that the responses of certain aspects of the image tend to fade away. But what we found is that the same patterns of connectivity also tended to support in our models synchronized oscillations. So that, for instance, if you have two edge detectors that are on a trajectory that lets them be co-circular, that they're more likely to oscillate in a synchronous way. Now, the beauty of that, as opposed to a system in which you have to relax, is that it doesn't take any time. You can detect a synchronized oscillation essentially at the first spike, because it means that that all these spikes are arriving simultaneously, and you can detect that, and neural systems are very good at detecting that. But what it says is that now we have two different implementations of the same basic functions. One appears to be, they both seem to accomplish the same things from a high-level performance point of view, but one seems to be more neural, both in the nature of its representations and the fact that it allows us to make these discriminations very, very quickly. So we actually extended this for fun, and we developed ODD kernels to try to discriminate between dogs and cats. And I think we probably did about as well as Google, although I haven't tried to compare it in any detail sense. So we actually found that to be very useful. Recently, we found something even more useful. This was arguably a consequence of something that DARPA did a few months ago called the Innovation House. Basically what they did is to put a bunch of people, or actually several small teams up in a hotel in Arlington and say, solve this image processing problem. And so we had a team that was led by my principal collaborator and co-conspirator, Garrett Kenyon, who took a bunch of graduate students and other people who were not tied to to family and home and so forth, took them to Washington, set them up in a hotel, and attacked the problem of looking at overhead video. And the thing that they focused on was something that that depends on local computation. And part of the reason for doing this is because we had already come to appreciate that in both GPUs and CPUs, the big problem for dealing with spiking is shifting weights in and out of memory. That is the bottleneck. Now there are hacks that you can do to make it work efficiently enough so that you can actually make progress. But ultimately what you really want to do is to put the memory and the processors right there next to each other, maybe even let the memory do the processing as people have described earlier in this meeting. And the question is, what kind of algorithms can make and take advantage of that kind of architecture, and what can you do with them? So Olshausen and colleagues, and I should say that Olshausen initially came to our attention with this description of the power of sparse overcomplete basis sets for describing and of learning these from inputs. But recently they had a paper that I guess Rosell was the principal or the first author. But what they described as locally competitive algorithms that can do computation. And so these nodes in this algorithm are essentially representing dictionary elements, and they fire. It turns out that firing is, while it's not essential, it turns out that neural dynamics are essential for this algorithm to work. So you can fake neural dynamics with things like Hopfield dynamics, but you have to have some sort of dynamics in the system for this algorithm to work, and spikes work extremely well. So what happens is that that this system, if a representation is activated and driven by input from the external world, what it tends to do is to suppress all the surrounding basis functions. So what it tends to do is to drive you into a sparse representation. But if there is some other basis function that is activated, which is not significantly overlapping with that first basis function, it can also remain active. So what it tends to do is to suppress the redundant information, but it allows the things that independently describe this pattern to persist. And it turns out that one of the things you can do with this is you can very efficiently, in essentially a single cycle, compute an L0 norm or an L1 norm. And for those of you who've ever tried to solve certain kinds of inverse problems, it turns out that that's a very powerful thing to do. So, for instance, in my case, I spent a lot of my career trying to determine sources from MEG data. And if I had had an L0 norm, I would have made a lot more progress, 15 years ago. And it's certainly my intention is to go back and apply this kind of neurally inspired algorithm to developing sparse representations of sources that can account for surface magnetic or electric field data. As an aside, the fact is that we can have a spiking neural implementation of these locally competitive algorithms, and they have some real advantages. For one, and this is the one that's kind of highlighted in this slide, if you have continuous temporal data, so if you're looking at video as opposed to single images, it turns out that locally competitive algorithms are very good at having representations that are consistent across time. So once a basis function appears as part of the description of an image, it tends to persist. It has inertia. And that's a good thing because, in the real world, things don't morph. If we see, an object at one instant in time, chances are at some other instant in time we're going to see that same object. It may be translated or rotated or changed in some way. But this persistence and this compactness of representation is actually a very important advantage. And it also turns out to be an advantage for things like solving the inverse problem for MEG, where it's very difficult to interpret any given map at a point in time, but if you look at it across time, you can actually come up with a compact representation. So, so moving on. One of the things that we found to be very difficult is that if we want to exploit this kind of temporal structure in visual imagery, in the representation of visual imagery, it's important that you somehow get that temporal structure in. And it turns out that it's very difficult to introduce appropriate temporal structure of the sort that I described earlier at the stage of visual cortex. If you start out with a retina that's encoding information as Poisson spike trains that are just frequency encoded, then trying to get this very structured kind of representation at the level of cortex, darn near impossible. You can do it with tricks, but there's no reason to think that the tricks you're using are anything like biology. On the other hand, as we say, this kind of temporal structure actually gets naturally introduced at the level of the retina. So one of the things that we thought about is, so what is it that drives this kind of synchronized oscillatory activity? What is there about the retina that tends to do that? Well, it turns out that there are network resonances, and so there is a tendency for the retina to oscillate at something like gamma band frequencies, and that's part of the problem. But it also turns out that in the eye, even if you try to fixate, the eye is constantly moving. That's probably a good thing because the retina is actually a lot more sensitive to temporal changes in its input than to simply static spatial structure. But it also turns out that these very fine movements in the retina, these so-called fixational eye movements, actually operate at about the gamma band. They're just about at that frequency. And one consequence of this, as you can imagine, is that if your eye is moving and there is like a fixed edge in your environment, as your eye sweeps across that, what it's going to do is to produce, because of the physical nature of the stimulus, it's going to produce a synchronized firing along that edge. And I would argue that, in fact, that is the thing that is at least part of the trigger for these synchronized oscillations that seem to encode metadata about the visual world. So we did some simulations of this. We started out with things like Gabor or sinusoidal gratings. Then we simulated movement. And one of the things you can convince yourself very early is that if you have movement along this axis and you've got a noisy grating, it actually improves the grating because it gives you an averaging effect that works for you. But if you have movement in this direction, it actually just blurs out the contrast and you lose the ability to determine the orientation of the grating. That's if you're encoding with rate. And so you're trying to extract information from the integrated firing rate. But if you encode with synchrony, it turns out that all the movement works for you because the movement encodes or causes you to have these synchronized patterns of firing that then you can extract with neural circuitry. So, so moving on and into the home stretch, this really motivated us in a lot of ways that we had to deal with the input functionality. We had to properly encode information if we're going to use neural systems to extract it, or neurally inspired systems to extract it. So we developed dynamic models of retina that are trying to reproduce the spiking function. Now there's a lot of other stuff that the retina does, things like enhancement of contrast and so forth, that are really important for its function. But we're going to neglect that right now and really focus on what the retina does in order to produce the proper dynamics in the output stream so that the rest of the system can work with it. Now this is a little movie that we made of the representation of the output of retina for a very simple kind of structure. Let's see if I can make this thing go. so this is a representation of the spikes that that you see coming out over the optic nerve from this fairly sophisticated retinal model. Now, if you look over in the right-hand side, what you have is essentially integration over time of those spikes. And what you can see is that integration over time gives you a very good representation of the structure that was there, right? These are obviously squares. Now this one was moving, as you may recall from the video, and so it sort of blurs the leading and trailing edges, and that's again because of the integration. You're only integrating, you're kind of partially sampling that edge as a function of time. But if you actually look at a typical spike frame, actually doesn't have nearly as much information as you might have imagined. It's kind of hard to argue that there really is any box down in the lower left-hand corner. And that's because it's really a combination of the rate and the synchrony that allows us to encode and extract that information. But even more interesting is that this model shows that that although responses to both of these boxes are synchronized and both of them are oscillating, that they're actually at slightly different frequencies and at slightly different phase. And what this means is that the system has encoded these as separate objects, even though it's using the same mechanism to encode them. I think that is, in fact, a key to how the system actually represents information. So, so most of what I showed you previously has been in software. We've developed an even more simplified version of the Retina that we can run on an Fpga. We're obviously not the first to do this sort of thing. There are some very nice examples of image processing engines that use FPGAs to process visual information. To my knowledge, this is the first that actually produces a biologically inspired spiking output train, which is clearly the motivation that we had. One of the things that we learned early on is that one consequence of even these very simple models is that because neurons tend to respond first to the highest contrast elements in the image, it turns out that the first volley of spikes that you receive from a retina like this really encode the gist of the information. And this is important because that means that you can resolve things based on a very small amount of time. You don't have to wait to integrate for 50 or 100 milliseconds in order to get the idea that the key idea comes through. So if you watch closely, I think you'll see that the first frame really gives you, the cartoon really gives you the sketch of what's going on here, and that the other stuff fills in the details. So we can take this idea further. We can build Fpga neurons. What we find is that if we build neurons that have what we consider to be the adequate degree of complexity, even for leaky integrate and fire neurons, we can probably only get 1,000 to 5,000 of them on a pretty hot Fpga. Now, that's a problem because we obviously think that we need billions and billions of neurons in order to do the kinds of things that we're doing. I think there are a couple of ways that we may be able to solve this problem. One is that in the visual periphery, you could argue that the same set of basis functions might be used to represent essentially all of the patches of visual field that you might want to represent. And so it means that the same circuitry could be employed, and the fact that these circuits are much faster than biological circuits means that you could temporally multiplex them over the whole system and use them in that way. So if we build, say, a little pipeline that leads from maybe a 9x9 or a 3x3 or some small patch on retina, run that up through a series of cortical columns all the way out to some visual area, then we could apply that same pipeline to process a number of say, overlapping patches on retina. It's also possible that if we want to simplify the process further and deal with arbitrary things that neurons can do, we can store the synaptic weights off board, use the FPGA as essentially a convolution engine, and flow those weights through as a vector that allows us, and essentially only send the weights that we need, because a small minority of the neurons, the input neurons, are firing at any given time. So we can still deal with large systems, but essentially by flowing the data in as a vector from off-site and using it to make sure that we get the optimal efficiency. So where are we going with this? We believe that one of the key places where there are important advantages for this kind of system is if we want to have deployed systems, if we want to have essentially a visual cortex that lives on a UAV. What we are imagining is that instead of taking a camera and bringing it into a cluster of cells or a cluster of computers that have GPUs and that sort of thing, if we could build a very efficient system for doing this kind of computation, we can get to, I think, the kind of power requirements and performance requirements that we need. So the approach is to use spiking systems, to use sparse over complete dictionary, to use locally competitive algorithms to compute sparse representations, and this produces a lot of magic, or so we argue. The specific things that we are working on these days are to build a spiking retinal processor that can operate in real time, that can give us millisecond types of temporal dynamics, to use those kinds of spiking models for spatial temporal inference, and by that we mean doing things like segmenting objects and doing things like tracking objects and predicting where they're going to go. As I say, I think locally competitive algorithms are an example where there are significant applications outside of visual processing, and that's something that we're pushing toward. Using FPGAs to implement neural processes, I think, is a workable intermediate step. Ultimately, I think that neuromorphic systems that are even more specialized will probably prove to be the winners. This is an example of a neuromorphic system that represents a million neurons that was developed by Gert Kauenbergs and colleagues at UCSD. And ultimately, I think that things like crossbar arrays, such as we have thought about and our colleagues at the University of Michigan have proposed, will be the implementation method of choice. Finally, just wanted to say that obviously this is work that's been developing for a long time. I'd like to, in particular, highlight my collaborator Garrett Kenyon, who has been my principal confidant and co-conspirator over the last 20 years, probably. And I also wanted to highlight one of our students who's here, Dylan Payton, who's made tremendous contributions to the work over the last couple of years as a graduate research associate. The last thing I wanted to say is that unfortunately, even though living in New Mexico is a great thing, and I've actually really enjoyed my career at Los Alamos, the downside of it is that my bosses expect me to be there for a meeting this afternoon, and so I'm going to have to leave shortly after this, but I would urge anybody who wants to talk about this more to send me email, JSG Juliet Sierra Golf at lanl.gov, and I'd be happy to continue this conversation. Thank you very much. So, any questions?
We were showing those co-oscillating neurons. That doesn't necessarily mean it is a study timing, right? That still could be a rate code, right? Or neurons.
No. Yeah, one of the points I was trying to make in that first little discussion of the physiology is that that oscillation is actually one of the things that happens when you drive neurons into a highly stimulated state and maintain it for a while. And that clearly can be a rate code. The kinds of things that tend to lead to synchrony, I think, are different. So I would argue that while oscillations are something that support synchrony, for one thing, by acting as a local clock, that it's really the synchrony and coincidence that's more important for encoding additional information. All right, thank you very much.